Hi everyone, welcome back to my channel. My name is Cha Cha, and I'm a student who's also studying for the MCAT. Today I'm going to be talking about behavioral genetics, which is probably more in depth than you need for the MCAT, but I'm also have a midterm for this class, so I decided to make um a YouTube video since why not with all my notes. And there's a lot of important stuff in here that's also relevant to the psych section of the MCAT, as well as for the biology section of the MCAT. So without further ado, let's get into it. So there's like some basics that you need to know for genetics, so that there's four distinct DNA bases. Um, there's adenine and thymine. So in RNA, which is a distinction, you'll have uracil instead of thymine, which will bond to adenine, but there will still be two hydrogen bonds between the two bases. Whereas then for there's also cytosine and guanine, which have three hydrogen bonds between them. So um, cytosine and guanine, because they have the three hydrogen bonds, it's going to be a stronger connection than adenine and thymine. So that's important. If you see like more C's and G's, that means that it'll have a higher melting point than if it's just a bunch of A's and T's that are in the string, which I've seen on practice problems. At least that's something that they like to try and trick you on. And it's because of the hydrogen bonds. You also inherit one of each chromosome from each parent, but we have a total of 23 distinct chromosomes. So we have 46 total, but 23, but there's overlapping of both. So we are diploids, which is sort of what that means. So ploidy is like how many of each distinct chromosome there are. So for example, you have two copies of chromosome one, but chromosome one is one distinct chromosome, if that makes sense. So we have 22 autosomes, which are the like non-sex chromosomes. That's why I like to think about it. And then we receive two sex chromosomes, which determines our biological sex from our parents. X and X will be female, and X and Y will be male. And then the Y chromosome typically does not carry a bunch of genetic information in it, whereas the X chromosome, if it's two females, a lot of the times there will be X inactivation, which means that that chromosome will get turned off early on in development. But that's typically why you see like calico cats, for example. Oh, they're only female because they have X inactivation. That's how they get their distinct colors, is that certain depends on which X is turned off. And then it goes from there. So some transcriptional basics. I have this crude image. So you'll have an enhancer region, which is pretty distal from the promoter region itself. So it's very far away. So it's like a far, and then you'll have activators, which will bind to the enhancer. And those will attract mediator proteins. And those mediator proteins will have transcription factors. And transcription factors are what bind to the TATA box in the promoter. So a TATA box is like a sequence that's typically around like 30-ish base pairs away from where RNA polymerase will bind and start transcribing said gene. And but there, how does the enhancer kind of like, since they're without the DNA binding protein, you'll just have the enhancer will be down here. So I'll just have an E for enhancer. And then you have the promoter, which is down there. And they're like usually pretty far apart. Like they could be like 1,000 base pairs apart. How does it get from the straight line, or give or take a straight line, to this bent shape? It's with that DNA binding protein. So there would be a DNA binding protein, which basically forces the enhancer and tata and promoter sequence to be kind of like close to each other so that this can actually happen. So transcription factors can bind. And then they'll attract RNA polymerase 2. It's very important. It's RNA polymerase, it's not DNA polymerase, since you're forming mRNA. Typically, there's also different types of RNA, but messenger RNA is what will get translated by ribosomes into proteins. And there's also there's other proteins too, like DNA helicase is what unwinds the base pair so that the RNA polymerase can transcribe the parent strand. And then, so there's going to be different when you have a gene, there's exons and there's introns in said gene. And that's what codes for the protein. So it's not removed by the spliceosome. And the spliceosome is essentially what kind of rearranges exons and introns. And an intron is what's removed, not from the intron, removed. So that's where there's not going to be like encoded for the protein. So those nucleotides will not be part of the final product, those amino acids. And what do I mean by that? So I feel like talking about the spliceosome and essential splice sites is easier to have with an example than just like me talking about it. So for example, let's pretend that we have like these two distinct regions of said gene or the open reading frame. And I just like color coded it. So 1, 2, 3, 4, and 5. So it really depends on like the cell and what is like their signals, environment surrounding. That's kind of like what determines whether or not something is an exon versus an intron for said gene. But I also, before I like get too far into this, I want to first talk about the 5' methylated guanine cap, which is added to mRNA to prevent degradation and aid with translation. So it helps the ribosome bind to the mRNA, which is in the cytoplasm. And then depending on whether said most of the amino acids that are transcribed, whether there's hydrophobic or hydrophilic, if it's hydrophobic, it'll go to the rough ER. And the rough ER is rough because it has ribosomes on it, whereas the smooth ER is what helps create fats. Well, that's not really something I want to go too far into in this video. But yeah, that's what helps the ribosome bind in the cytoplasm. And then the 3' poly A tail is added, which helps. So it's just like a bunch of like adenines which are added at the 3' end of the mRNA sequence. And that helps with stabilization. And it is sort of like a postage stamp for mRNA to go to the cytoplasm so that mRNA can bind and the protein can be created. So it's like a postal code. So you need to have the postal code in order for the mRNA to get to the cytoplasm, right? Just like with any letter that you're going to send off, you need to have a stamp. Otherwise, how does it know where it's going, where it's supposed to go? Otherwise, it just kind of sits in that mailbox and the mailman's like, hey, you didn't pay for postage. I can't deliver this to where it needs to go. So now let's kind of talk about splicing in the spliceosome. So as I was saying, I have like 1, 2, 3, 4, 5. So these are just like different areas, but they have an essential splice site. That's kind of what marks the difference between 1 and 2 and 2 and 3 and so on. In this essential splice site, as I was saying, it depends on really like the signals and that that particular gene or that cell is in that'll determine what exons will be included or sorry, cut out and what exons will stay in and what introns will get cut out. So for example, I have 1, 3, and 5 is this is a protein isoform. Isoform is just like a fancy way of saying like a different type of protein encoded by the same gene. And then we have 2 and 3, 2, 3, and 4 down here. And as you can notice, that 3 is overlap. So you could have like depending, I guess, like how much like of these essential splice sites there are, you could have like infinite amount of these proteins, like these different combinations. You could reuse it for different protein isoforms too, like for 3, for example. And why? Like, why is this happening? Like, what's the point of having these essential splice sites? So all cells, except for red blood cells, they have the same DNA and chromosomes. But as you can notice, obviously different cells have very distinct functions in your body. Like you have like your immune cells, you also have neurons and stuff like that. So in order for them to have such distinct abilities, you need to have like these protein isoforms or just like these proteins in general, which are turned on and off at different areas, or these genes are turned available for transcription in different areas. And also isoforms are another layer that help make the cells unique and the proteins unique. And it also increases diversity too. Just like in general, diversity is always much better because the more diversity you have, the more likely that eventually nature will have the right combination for the genes to get passed on to the next generations so that species won't die out. And then I also kind of hinted at like an open reading frame, and then which is like where the genes are located. So there could be multiple genes located in the open reading frame. And there's also intergenic. Also genes, like they can overlap too, um, depending on where they are in the chromosome. And then we have intergenic regions too. So intergenic are the inter, meaning between the genes. And this is where most, 75% of your DNA are intergenic regions. So most aren't open reading frames. And then also variants are what, sorry, variants don't necessarily mean that's going to be a mutation. So variant can be a base pair change, but that doesn't necessarily mean that there's going to be a phenotypical change in the individual that has that variant. So for example, like if you have like an adenine change to guanine, it might not necessarily like result in a phenotype change. So like let's say if you have an adenine, you'll have brown hair. If it changes to guanine, you also have brown hair. But it's also very likely that there can be a mutation, which means that in the phenotype, that's what mutations are pertaining to the phenotype. And variants are talking about the genetic code itself. So it's very also you maybe that adenine is for brown and that guanine, if you change it, will be for blonde hair. And then the open reading frame, I didn't mention it's like large regions which are uninterrupted by stop codons too. So there can be multiple genes, as I said. And then these are exons are 1.5% of all variants. Or like not variants, but like just like DNA. I don't know why I said variants, but that's where like most of 75% of your like DNA is intergenic, and 1.5% of your DNA is exons, 25% are introns and exons. So now I kind of want to get into epigenetics, which is more of that regulatory process of which genes are expressed where and how we have so many different types of cells in your body, except for red blood cells, which are weird if they don't have DNA, as well as for mitochondria, since red blood cells are the transporter for oxygen. So it doesn't make sense for them to have mitochondria since mitochondria use so much oxygen. Um, so I want to talk about histones. So histones are proteins that DNA binds to. They are octamer. So octamer, like whenever you see like typically think eight, like an octopus, right? And DNA wraps around the histone twice, so it's 160 nucleotides around each histone. And the nucleosome is the individual subunit um, of a chromatin, which is essentially what like condensed or I'm not condensed, but like it's what DNA and its associated proteins are. So like you'll have a chromatin loop. So that's like where you have a bunch of DNA with its histones that are like interconnected between each other. So that's like a very regulatory, like this is the regulatory unit of your DNA. That's how you don't have all like 3 billion nucleotides in your body not expressed at once, since that would overwhelm your body. And 3 billion nucleotides is a lot. So you would probably die, or you'd have to be like a billion feet tall if they're all expressed at once. Euchromatin is loosely bound form of chromatin. So I think of like like euchromatin, it's like easy going, right? It's not super tight, like high strung. It's like loose, so it's available for transcription. Whereas excuse me, heterochromatin is tightly bound and it's not available for transcription. So it's highly strung. It's heterochromatin H for highly strung. And now we could get into more of the regulatory regions without changing the DNA. So there's acetylation, which is where an acetyl group will bind to a lysine, which I drew here, that H3 plus, with it's a lysine an amino acid. It's one of the basic amino acids, so it has a pKa of around 10, give or take. So meaning it's basic. And so lysine is otherwise positively charged, right? So if you have an acetyl group that binds to lysine and it gets rid of that negative charge, then that means that the histones aren't able to bind to the DNA anymore. Since DNA has a negatively charged phosphate group, So the lysines and the histone, right? Like the protein, it could bind tightly to the DNA, and that makes it unavailable. So when you acetylate a protein, when you acetylate the histone, then that means that your DNA will be able to be transcribed. So it's in its euchromatin state. Whereas for methylation, so there's two different types of methylation. So there's methylation at the histone level, and this can either inhibit or promote transcription. It depends on like which amino acid that you're adding the methyl group to. Whereas at the DNA level, it inhibits transcription. So if that means it'll be in its heterochromatin form, right? So this occurs at the nucleotide cytosine. Cytosines before or pre-guanine. So CPG, that's like the acronym that you could try and remember. So it's CPGs are enzyme recognition sites, and they're also typically like called CPG islands. So there will be a bunch of CPGs typically like right by the promoter region, which makes sense, right? Since promoters are what starts to encode uh ge- I'm not code, sorry, transcribe genes. So it makes sense that they'd be by the promoters, just because like as close to the action as possible, as closest to inhibiting that RNA polymerase as it needs to be. So yeah, I already mentioned this, like variants are what change the code itself, the genetic code itself, whereas mutants are when the phenotype changes. And I could write that too. Mutants change in phenotype. Okay. So let's kind of get into the different variants that you can see inside of your body. So there's synonymous mutants, not mutants, sorry, synonymous variants, sorry. That's when your base pairs change, leading to the same amino acid. So so for example, like going back to that adenine to guanine change, how the heck can that lead to the same amino acid? So amino acids are degenerate. So multiple codons can lead to the same amino acid. So amino acids, so a codon is 3 nucleotides. And that means like um so you have 20 amino acids, right? But technically, like because it's 3 nucleotides, you could have 64 possible codons. So obviously there's going to have to be some that are reused. And that is true. Also, there's stop codons too. And stop codons are the only codons that will not be encode for an amino acid. So the start codon, which is UAG, that will have methionine is always for the start, and then the stop codons. There's 3 that encode for nothing. No amino acids. So what do I mean by a synonymous? So like let's say you have the cat drank milk. Like that's like your sentence. And then you're gonna replace the C in cat with a C. Excuse me. And then there's missense mutations. And that's when the base pair change leads to a different amino acid. Um, so for example, I had the cat drink milk up there, and now I have the bat drink milk. So typically missense mutations, they aren't super loss of function, but they can be. So for a common example of this is cystic fibrosis, which is an absolutely devastating condition where you have chloride ionotropic channels in your lungs, and those aren't working properly. So it can lead up to a bunch of like flu up, or edema, which is like swelling. And this is all, yeah, it's really bad. It's really painful. It also makes you like a lot more susceptible to infections too. But it's all because there's like a deletion, or I guess I shouldn't say like it's a missense one, but it's like a deletion of phenylalanine at position 508. So that's just like a single amino acid change can lead to like this horrible disease. So I shouldn't say this is cystic fibrosis. I don't know why I put that here. That should be yeah, it's a deletion, I guess. It's a what it is instead. So my apologies for getting that confused. the point still stands. Like that phenylalanine is like a key hydrophobic raft in the protein. So that chloride channel can't um form, and then chloride itself, right? Like ions and through the power of like osmosis to like that's how water flows throughout your body is when you have high concentrations of ions is how water flows. So if you don't have that, then water can't flow properly, and then that's how you get that swelling. Um, so a nonsense not mutation variant is like when there's a premature stop codon. So you can't read all of the code. So this is rare to see in a human because it's typically so deleterious and loss of function that a lot of times you won't survive a nonsense mutation. Um, so going back to my cat example for the cat drank milk, instead of like having the cat drank milk, it's just the. Like there's not any more of the sentence. So how are you supposed to make sense of just the? It's just a word. I kind of think about like when you have like a really fancy like in a storybook how they have the T and that's kind of how I think of like nonsense mutations. Or there's just nothing really afterwards. You're just like so distracted by the you can't see anything else in the story. Frame shift variants. You insert or delete a nucleotide not in a multiple of 3. So this is rare, very deleterious as well. So you won't see this often in humans because this a lot of the times they won't survive or the embryo is not viable. So for example, like going back to the cat drank milk example, I added the L to cat, so the za like that can still be read the same, like that makes sense. And then I have the claw, chur, dra, camo, k. So that doesn't really make sense after there. It's complete nonsense. And then I have insertions, which is where you insert nucleotides in multiples of 3. So, excuse me, you can still make sense of the like series of words, but it may not make a ton of sense. So if I have like the fat cat drank milk, it's just like something extra. It could be loss of function, and it could also be gain of function too. Whereas deletions, same thing. That's where I was talking about that cystic fibrosis option, where it's the like cat drank milk. And then so that's where I got rid of the. So it's just cat drink milk. You could still make sense of it, but it's not as cohesive. Same with the fat cat drink milk. Like you're just adding extra stuff. Whereas this case, you're getting rid of something not like super important. But let's say if I got rid of the cat instead of like the drink milk, that doesn't make sense. Or if I were to add a word here, if I were to say the human cat drank milk, that doesn't make sense. So there's two different also types that I want to. Oh, I guess let's talk about short tandem repeats before I go into the uh RNA. I let's talk about short tandem repeats. So they're repeating a small amount of nucleotides. So typically more or less than what is typical in the humans. So we have a bunch of short tandem repeats. It's normal to have short tandem repeats. So for example, like Huntington's disease, you won't have Huntington's disease as long as you have the CAG repeat less than 39 times. If you have it more than 39 times, then you'll have Huntington's disease, which is typically it could be fatal after I think like most of them don't reach adulthood. And then there's also Fragile X syndrome, which is when you have greater than 200 repeats of CGG in a promoter region of the FMR1 gene, which leads to hypermethylation. And we were talking about methylation earlier and how that can be devastating. Another devastating, it could be another regulatory reach of histones and DNA. So leading to the gene being either improperly turned on or not or turned on too much. And then we have siRNA, which is like where specific targets it targets specific regions of mRNA. So they are very exact in their targeting. and they silence it. So this is after like you already did the whole transcription thing, and then boom, like the mRNA, like it binds to sRNA, it binds to the complementary region, and then it's silenced, so that protein won't get transcribed. A lot of the times, this is referred to as a knockdown mutation because there still can be some mRNA of that protein that's through the cracks. Whereas a complete knockout would be if it were at that transcriptional level, like where the mRNA isn't even transcribed. And then we have mRNA, which is where it binds to the 3' UTR region of mRNA, and that silences and destabilizes the mRNA. So it's also another version of a knockdown. But this is just stuff that your body naturally creates, or you could also, a lot of the times in research, people will have, they'll design probes that can go into your body and it can knock down certain specific sequences of your mRNA. So I'm gonna talk about now like how rare are variants. So we share 99.7% of DNA, which is kind of crazy if you think about it, just because if you were to look at people, they look very different. Like for example, like I am white and I have like brown hair, but I look very different than my best friend who has darker skin and brown like dark brown hair. So race is a social construct. It's always changing, like especially if you look at the census throughout the years, we're adding more and more races than compared to like 100, even 20 years ago. However, the allele frequency does change across the ancestry, especially. So there's actually the most diversity in Africa just because that's where humans originated compared to like after you spread out from Africa, like to compared to like the US, for example, or North America. Excuse me, hold on. So of the 0.3% of DNA that we differ in, 95% of that DNA is like common mutations. So it's like mutations that are like spread throughout uh the world, but everyone, it's not like region specific. And it's also just like, for example, like why we have like blue eyes versus like brown eyes, for example, that would be a common mutation. But the 0.3% is still 4 to 5 million nucleotides that we differ between everyone else. So we have around 3 billion nucleotides, which is crazy. And also, I want to talk about reproductive fitness too. So that's when you pass on genes to the future generation. So it's like if you have like housekeeping genes, which are genes that are like essential, so they're turned on in majority of the cells. If you have a loss of function mutation in one of those housekeeping genes, it's unlikely that that embryo will be viable, which is what I was talking about a little bit earlier. Then also we have a total of around 20 loss of function genes just in general, like which is kind of crazy to think about since we talked about how detrimental loss of function can be, but there are certain genes that aren't imperative for our survival, so we can still live past to adulthood. Then I also want to talk about de novo mutations. These are new mutations, although there are lots of checks and balances, like when we're like encoding for stuff, like when you're like first transcribing, there can still be new mutations. These can be either rare, where that embryo won't survive until adulthood. So for example, like if both of the parents are like totally healthy, they can also have an embryo which isn't healthy. Like let's say, for example, like it has like a heart murmur. That for unfortunately, that embryo is not viable and it'll die in utero. But there's also mutations that can be beneficial as well. Not all mutations are bad. Just a lot of them are. But the mutations that are good, a lot of the times they'll live to pass the they'll live to adulthood and then they can pass on their genes to their children if they choose to have some. Um, sorry, I'm like sick. I don't know what the heck. I've been sick for like a week and a half, but grind never stops, I guess. So I want to talk about like the types of genetic studies. This is not imperative for the MCAT. I just think like these types of studies are interesting. But first I want to talk about like the different types of like mutations, like a broader scale, or not even mutations, they're like variants. So you have like single nucleotide polymorphisms, which are also called SNPs, and that's when a single base pair can cause changes. And they can have small or large effects. These are majority of variants that are inside of your body. So when I was talking about that 0.3% difference, most of them are SNPs. They're oftentimes not in genes too. A lot of the times they're in energetic regions. And then an indel is less than 50. This is like a larger scale variants, but they're less than 50 base pairs. Whereas a structural, typically that'll have like a much larger effect. That's when it's greater than 50 base pairs. So there was something like this was typically popular in the 90s and the early 2000s called candidate gene studies, and it is now out of favor. Like if you see someone that's doing a candidate gene study, like you should kind of like side eye them, like roll your eyes and be like, how are you credited as a scientist? Since a lot of these ignore the intergenic regions, which is like as we talked about, that's like a huge portion of our DNA, the non-coding regions, like not in the open reading frame. And it also ignores polygenic nature of most genes. Like, for example, most genes are very complex. Like, even for something like eye color and hair, like, that's not going to just be one gene, unlike what we were taught in like your very introductory, like, biology courses. Like, maybe when you're doing that Punnett square. There's also lots of false positives, because there's too large of a p-value. Like when you're looking at something that has 3 billion base pairs, right? You should probably have a very small p-value instead of like even 0.01. That's not good enough. Like that doesn't necessarily mean that there's like any correlation there. And then it also looked at specific genes that researchers suspected were like leading to said variant or like leading to a phenotypic difference, that mutant, which is also not something that you should go into. You should kind of like, especially for like gene studies, since you're looking for stuff that may not necessarily be there. It's a confirmation bias. So what is that being replaced with? So there's something called gene-wide association studies or GWAS. They are now like that's a new trend. Hopefully we'll see if something better will come along. But that would suck just because spent a lot of time on this and also means like some of the results aren't credible anymore. But unlike candidate gene studies, which weren't replicable, gene-wide studies have been, or GWAS studies are. If they are correlational, they are not causal. Just because like SNPs, like you can't attribute a SNP to a gene since a lot of them are in energetic regions. You also have to consider the polygenetic um like nature of both genes, like I was talking about. So there's multiple genes that relate to one trait. So what is a trait that we're looking at? So a trait would be something like height, hair color, like narcissism, just stuff like that. I guess like intelligence, like just stuff that are like really complex. And it's also, yeah, so as I was saying, it looks at SNPs. So I have a Manhattan plot, which is just kind of like an example of it. So its threshold, like I was saying, like up for candidate gene studies, it was 0.01. But for GWAS, it's 5 times 10 to the negative 8. That's crazy. Like that's so small. But so that's how you know that there's like a good chance that there's probably not too many false positives, which is kind of, or at least so far it's been true where most of these studies have been replicable. But this also means because you have 5 times 10 to the negative 8, you need to be looking at a ton of SNPs and you need to have a lot of people, like 50,000 people. Like most of these studies are like 50,000 people. You also need to have a large and diverse sample. So you need to avoid population stratification, which is basically what I was saying, where it accounts for when people have there are different regions of the world have different allele frequencies. And there's also different variant types that are more common in different parts of the world than in others. Like Africa is more diverse. Like that continent is more diverse than North America. And then also there's a lot of like regression and analytical techniques, which is like Plink, where Plink synonymously, not synonymously. simultaneously runs a bunch of regressions on those SNPs just to make sure that that's to that 5 to 10 to the negative eighth p-value to see if there could be some significance. Yeah, there's also fine mapping, which is another technique that a lot of GWAS studies use. So they look at where those SNPs are. So they look at like kind of context clues. Like, is there a ton of like acetylation that's happening in that area? Could it possibly be an enhancer, a promoter? So they look at stuff that's like common characteristics of like different areas. Like, for example, like a promoter region, like it'll have like binding site, or it'll be like a TATA box. There will be like transcription factors that'll bind to it, as well as RNA like polymerase will be like nearby. Just think about like what it could impact if you were to have that step. And then there's something called eQTLs. And that is like when you look at how different SNPs impact gene expression. So we're looking at gene expression, not trait type. So we're looking at the actual like dosage level, whereas for GWAS, you're looking at the trait type itself. And then typically like eQTL, since you're looking at dosage types, they're within one MB of a promoter, which makes sense, right? Since promoters are what control like gene transcription. And then some like further analytical techniques is there's microarrays, which is when you have hybridization between target genes and fluorescent probes. So it can indicate like expression levels, like depending on like here's a microarray here. That's it can tell you like, hey, like these dark areas, that means that that gene is not being transcribed. Like there's no it didn't bind to our complementary probe, so therefore it's not transcribed. And you can look at thousands of these at the same time. Whereas if you have allele-specific oligonucleotides or ASOS, it's highly specific for mutations or variants that are even one base pair difference. So, and you can kind of measure that with the fluorescence again. So you can see like if you have like this like area for that gene, like if you replace it with like the AA like homozygote, like that's a lot more like fluorescence over here, right? Then where the AC, they're kind of like both at the same time, like they're medium, like there's 50-50. So you have both of the genes, or sorry, both alleles, and then the CC homozygote, where you only have the one allele. Yeah. And then I don't know if I mentioned, but alleles are just like uh different like versions of the gene. That's just what an allele is. And then we have ACE models. Um, so typically this is looking at twin studies. So particularly monozygotic twins. So they come from the same egg, hence the mono being one, and zygote meaning egg. And then there's additive. So what is each of the ACE is an acronym. So what does each letter stand for? A is for additive genetics. So we're looking at allele effects. For that, it's not specific for that individual. You assume the twin has it as well. And you also assume that additive, like why is it called additive genetics instead of just like genetics? You assume that the allele effects build on top of each other over generations, so it's cumulative. So that's like where you have like your haplotype blocks. There's like areas that are like little crossing over recombination, which means that those specific genes or the specific base pairs, like they have really high language disequilibrium. So that means that they just like are like connected. Like they'll typically like they're like within they're really close by to each other. So they'll just like get transcribed together. Like they're like best buddies. They're like inseparable best friends. Yeah, that's when you just assume the mutations carry over. Then C is for common environment, so or shared environment. So like the SES is the same for twins, right? Like your twin isn't just like unless like something tragically happened to parents, like if they got adopted, like typically you have the same socioeconomic status growing up, same race, same family, same school, yeah, just like same neighborhood, etc. And then the E is for error. It's also like this is only one that's like specific for that individual themselves. So it's like when you have experiences that are unique. So for example, like maybe that twin, right? Like they probably have some different friends than the other one. Or maybe like they'll go to different colleges and then they'll get married to different people. Um, but this can be kind of like GWAS studies and just like genes in general. Like it can be kind of dangerous without this rhetoric of just essentially finding out like where our differences is. It can be weaponized, especially by people who want like to separate in groups versus out groups, which has been seen time and time again with especially with the Nazis. Then eugenics is what I'm talking about. This is like probably the most famous example of this, but unfortunately, there might be more of this that can happen, especially if like we don't protect this data and like if we feed into the rhetoric that we are different instead of the same. So eugenics is scientific racism. It applies a lot of Darwin's plate, like characteristics applied to humans specifically. So you're trying to quote unquote breed out the bad phenotypes. So Galton, Franz Galton, he was the founder of eugenics, and he had a lot of this like negative biasy. That he had positive eugenics was popular. So that's when you have increase in trait. You try and have people quote unquote breed, his words, not mine, for traits that are deemed desirable. So for a lot of the times during uh like just the time period they grew up in, hence like the Aryan race that came from Hitler, that was blonde hair, blue eyes, pale skin. Whereas the negative, you would eugenics is like when you tried to prevent people that were considered undesirable from reproducing. So specifically people with like darker skin, darker hair, dark eyes, just dark in general, which is not true. Um, and Linnaeus, he was kind of the first that created the ideas of races. He was a Swedish individual, and the idea of race, he also had like different connotations with said race too. So obviously, well, not obviously, but like given that time period, he had white people on top, and then like the darker your skin went, usually the worse the connotation was. Like the worse, most undesirable like characteristics that person had, which is obviously not true. There are good and bad people of no matter what you look like. The Nazis were so inspired by US eugenics in the 1930s that they like that's kind of how they adopted like a lot of their policies. The US was so appalled by like them taking it so far that that actually led to a lot of death of eugenics in the US, but that didn't really stop the South uh, from still kind of continuing forced sterilization. So there's around, I think, like 40,000, don't quote me on this, but like 40,000 individuals in the US that were forced sterilized. So they were allowed to have children. So either because of their quote unquote race or because they had some like mental health illness or religious purposes. And a lot of those times this was done at asylums, and disproportionately it was Black women, followed by white women, then Black men, and then white men. But for the men, it was very rare. But for Black women, it was disproportionately high. But this didn't stop, yeah, the South, they still continued this practice until like the 1960s. There was like the famous saying of like Mississippi epidectomy, which is a forced sterilization of Black women. So yeah, it's kind of crazy to think about that wasn't even 100 years ago. Which is why history is so important when you're considering biology. Just because like how can you think about we're always thinking about moving forward and having like better practices, but it's also really important to consider the implications of our testing as well and just how it could be weaponized. That's all I have on that note. That's all I have about behavioral genetics. Hopefully that made sense. I really enjoy this topic. I just like genetics and like general. I think it's really interesting. Not like eugenics as, but like just like the new stuff that we're learning, since I think that it could have a lot of like practical applications for health. Yeah, so let me know if I made any egregious errors. I already caught some of them as I was filming this video. So hopefully there's not too much more. But yeah, hope you guys have a good rest of your day, and I'll see you later.